Hey everybody! So I'm finally getting back to screencasting because I found my screencasting headset, but it seems I'm developing a cold, so I'm going to try and keep the sniffling to a minimum, but it may just happen anyway, so just warning everybody that that's a possibility, and I apologize for it. Okay, so um, obviously I have skipped the meiosis chapter, and that's because you just had an exam on it, and this is a little bit more important to make a screencast of in light of the upcoming final. So I'm planning on making a meiosis screencast, but that's probably going to happen on Friday or over the weekend. So let's get started with this one. So, the study of how traits are passed from one generation to the next is the study of patterns of inheritance or genetics. So, your heredity are your traits that you are capable of passing to offspring and those which you receive from your parents. So, I'm just going to get my pen ready. Pick a different color. I like this purple color. Okay, so your heredity are your traits that are heritable. So either you can pass them along or you receive them from someone else. So here we have a kind of a mean joke, which one guy is asking another guy, why the long face? And this long-faced fellow is saying, well, because I inherited it, aka it's not my fault. We shouldn't be making fun of people's faces. That's not kind. So one cool thing about heredity is that we can predict how likely it is that an offspring will have a particular trait, because science knows a lot about genomes and how traits are passed along now, so we can actually make predictions about those kinds of things. But to prove a point, here we have a spliced together photographic image of a grandmother and a granddaughter, and as you can see, although of course the grandmother is older and aging has taken hold, they have an extremely similar face shape and eye shape. So that's evidence that heredity is true, that traits are heritable and you get them from your parents and grandparents. Here's another couple examples, and these are a little bit more scary to look at. Oops. Why can't I just use my pen? Why is this so hard? PowerPoint. I just want purple and I just want pen. Can I write? No? Okay, well then. Sorry guys, I don't know what's going on with PowerPoint right now. I can use a laser pointer. Can I use the pen? Now I can. Okay, well. Sorry, that's very irritating. I don't know what the deal is. Alright, so father and son, you can see even though this image is a little bit freakier to look at, they have similar traits, and even father and daughter. Okay, so Mendelian genetics refers to the discoveries about the heritability of traits that are specifically ones that were discovered by this fellow, Gregor Mendel. So that's Gregor Mendel, and he was an Austrian monk, and he was performing these experiments at the end of the 19th century. So what he did was he observed that certain characteristics of pea plants seem to be able to be passed on to subsequent generation of pea plants, but not with 100% accuracy. So depending on which crosses were made, you would get a different assortment of traits. So what he resolved to do was basically say, okay, well, I'm going to count the number of offspring with each kind of trait, and I'm going to record these numbers in a ledger and see if I can find any trends in there. So he used probabilities, and these probabilities were derived from counting. So he didn't just receive them from somewhere, he calculated them. And doing this, he was able to predict the likelihood of a particular trait showing up in one or another generation of pea plants. So let's get into the details of that. Sorry, I had to pause there to cough. So he used pea plants to study the rules of inheritance, and he counted very carefully. So part of any good scientific experiment is the ability to quantify your results and make sense of them mathematically. So any good hypothesis should be able to be falsifiable, but also quantifiable. So you should be able to come up with numbers that describe what you're seeing. Okay, so a little bit of background about pea plants so that you understand how they work. Ordinarily, pea plants are self-pollinating, so they have both male and female sex parts within their flower, and they just go ahead and fertilize themselves to produce true breeding or identical plants or as offspring. So the female sex part of a plant is called the stigma, and the male part is called the anther. So ordinarily, this plant's going to self-pollinate, and then the seed pod will drop off, and the seed pod is the ovary. So the seeds in the ovary, these are going to drop off the plant probably nearby, and that seed or seeds will germinate nearby. And so this is how pea plants propagate. So that's good, but if you want to crossbreed, so let's say I want to breed this pink flowered plant with a white flowered plant, I would need to prevent the pink flowered and white flowered plants from self-fertilizing. So how do we do that? Oops. Additionally, there's a variety of traits that pea plants are capable of having that differ from other pea plants. So this doesn't mean they're different species. It just means that it's a different variety of the same species. So just as I have brown eyes and some of you might have blue eyes, it doesn't mean that we're different on the species level. We're still both Homo sapiens. But we have different traits, and these traits are heritable. So same deal with peas. So within this pea pod, we have some peas that are wrinkly and other peas that are smooth. And this is one of those traits that is heritable. All right, so let's look at self-pollination. Unfortunately, this process is actually pretty simple. If it hadn't been, maybe Gregor Mendel wouldn't have found what he actually found. So remember that within the flower of a pea plant, there are both male and female sex organs. So in order to prevent self-pollination, you're going to have to remove one or the other. So the easiest thing to do is to remove the anthers, which again are the male parts. And once again, Can I draw now? Thank you, PowerPoint. Okay, so the anthers are the male part of the plant. So these are going to produce pollen, which is the plant equivalent of sperm. So once you've done that, now the plant can no longer self-pollinate, but you've left the stigma, which is the female part, intact. So if you leave the stigma intact, there is still something available to be pollinated, so you can cross-pollinate by taking a paintbrush and dipping it in pollen of your choice, and then painting it onto the stigma of a different flower. So that's what Mendel did once he was sure that the original parental generation bred true. And we'll talk more about that in a little bit. Nope. Okay. So what did Mendel actually do? Well, he looked at 7 characteristics in his plants, and these were attributes or traits such as seed color and texture. So why did he pick 7? Well, that's the ones that he observed were heritable, and the ones where he observed there were 2 varieties or traits. So it wasn't the case that the trait was continuous like height is, where there are a variety of possible outcomes. He picked one where there was one version or another version, period. So short or tall, wrinkled or smooth, yellow or green, those kinds of things. So he observed which of those traits showed up in succeeding generations based on the characteristics of the parent plants. And that's really important because the parent plants contain the genetic substrate for the variation in offspring to occur, so you can be sure that the variation in offspring isn't happening for some other reason. So these are the actual characters that he studied. So these include seed shape, not, round or rhomboid, but rather just smooth or wrinkled. And seed color, so you could have yellow or green. The pod or ovary shape also changes, so it can be either inflated, so kind of puffed up, or wrinkled, where it hugs the peas, or the pod color can be different, so you can have either green or yellow pods. Flower color is also heritable, so you can get purple or white, as well as the position of the flowers on the plant and the length of the stem. So these are what he looked at. So now let's define phenotype and genotype. And this is something that students have a hard time understanding the first go around. So I'm going to phrase this in a couple of different ways to try and make sure that all learning styles are addressed as much as possible. So a phenotype is any physiological feature, bodily characteristic, or behavior of an organism. So the easiest way to explain this is that, let's use bodily characteristic. So for a plant, its body it consists of stems and leaves and flowers and whatnot, right? So we know that genes code for proteins. And proteins do the work of cells. So in multicellular organisms, proteins are critical to survival, and they determine what kinds of attributes the thing has, because most of the pigment molecules that give flowers their color are proteins, or they are derived from proteins. So let's say, for the sake of argument, that we are looking at a plant, a pea plant, and this pea plant happens to have yellow peas. So yellow is a pigment molecule in plants that is usually made of the carotenoid. Of course, I can write on the next page, but not this one. There we go. All right, so zeaxanthin. So zeaxanthin is a pigment molecule, and this equals yellow. So if the plant contains a gene for zeaxanthin, it will express that gene and the peas will be yellow. If it doesn't, it will express a different gene, or different allele, and the peas will be green. So gene for, let's call zeaxanthin Z, produces this protein, and the outcome of the gene coding for that protein is yellow. So yellowness would be the phenotype, and the allele, capital Z for zeaxanthin, would be the genotype. So phenotype and genotype are not interchangeable terms, but one does inform the outcome of the other. So genotype determines phenotype. And I used a physiological feature or bodily characteristic, but in animals with behaviors can be heritable as well, or behavioral sets. So we know, for example, that certain kinds of mental illness are heritable, and that would be an example of a behavioral alteration that was informed by an underlying genotype that was able to be passed along. So in Mendel's plants, purple flowers were one phenotype, and white flowers were another. So phenotypes are determined by genotype, which means the organism's genetic makeup, specifically which alleles of which genes the organism possesses. Oops. Okay, so beginning the experiment. So he started out with yellow and green peas. So remember, each of the characters he looked at had only two possible outcomes. You could have yellow peas or you could have green peas. You can have white flowers, you can have purple flowers. So these are two variations of the same character. So let's write that out. Color of peas. So the trait is color of peas, and the two options for that are yellow or green. So I just explained how you can prevent self-pollination to crossbreed, but if you are examining characters that come in one of two varieties, either yellow or green, for example, one of the things that needs to happen is that you need to be sure that your two parent candidate plants, so a plant with green peas and a plant with yellow peas, always produce that color of peas. That way you can be sure that the variation in pea color that you're seeing is non-random. So that's the first step that Mendel took. And this is evidence of him having a really finely honed scientific mind. So he recognized the value of ensuring true breeding in this first step. So true breeding just means that plants with green peas always produce green peas, etc. Once he was sure of that, he cross-pollinated a yellow pea plant and a green pea plant, and then he observed the offspring. So in genetics, we abbreviate the generations that result from crossing, just because it's easier to write P than it is to write parental generation all the way out. So P is the parental generation, and in this case we have a parent plant that always produces yellow peas, and a parent plant that always produces green peas. The F1 generation means first filial generation. So filial means son or daughter, it just refers to offspring. So F1 is the generation that immediately comes after the parental generation. So in Mendel's case, he crossbred yellow and green peas, and to his surprise, he got a generation of pea plants that only produced yellow peas, even though he crossbred with green peas. So that gave him pause. He said, that's peculiar. I wonder what that is all about. Then he allowed the yellow pea plants from the F1 generation to self-pollinate. So he didn't cut off the male anthers. He left them alone, and that created the opportunity for those plants to just pollinate themselves. And then after that, what he did was he cracked open the pods from all those plants and he counted peas. And he counted a lot of them. So 6,022 is a lot of peas to count. So in the F2 generation, he saw 6,022 yellow peas and 2,001 green peas. So if you were to divide these, what you would get is about a 3-to-1 ratio of yellow to green. Not exactly, but about. And the more times you do this, the closer the average will be to 3-to-1. So Mendel said, well, gosh, that's interesting. So the green trait went away for an entire generation, and then in the following generation, it popped up, but not in equal numbers to the yellow peas. So it wasn't a 50-50 ratio, it was a 3-to-1 ratio. So there must be something to this. There must be a reason why this is occurring. Sorry, I had to take a pause to blow my nose. Didn't want you to hear that. Gross. So let's move on. So Mendel observed this particular ratio, which means that something is going on. He's just not sure what. So what did he figure out from the experiments? Well, one was that the basic units of genetics come in pairs. So he didn't know the word gene or allele yet. He just noticed that whatever was causing the phenotypic changes and patterns he was seeing necessarily came in twos. So these units are today called genes, and they come in alternative forms called alleles. So let's remember our central dogma stuff, because all of this relates to everything else. So these things annoy me so much, but according to the internet, they are not fixable. So genes are regions of DNA that code for proteins. So you already know this because we've done some experimentation with observing how that process works. That's what the central dogma is all about. But what we haven't addressed yet fully is the possibility of having alleles. So alleles are versions of a gene. So let's give an example here. So for example, gene for pea color. So there's a single gene in pea plants that controls what color the peas are. So the character is pea color. The gene codes for pea color. You can have two versions of that gene. Big Y, that's one allele, and this is going to equal yellow, or little y, green. So those are your two options for that gene. So alleles are not on the same chromosome. So you inherit the alleles that you have for your various traits from your parents, and because, remember, you get one chromosome from your mom and one from your dad, and those chromosomes are said to be homologous. I'm going to underline that word. Homologous is another way to say the chromosomes contain same genes, different alleles. So one chromosome might contain a allele that is the capital Y, so that would be yellow peas, and the other chromosome might contain a lowercase y, that would be green. So another example is blood type. So if you are type AB, you get one allele from mom, let's say. So I just want to stress, it's not the case that moms always give the A-type allele. It depends on what mom's blood type is. So this is just an example. So let's say you get an A allele from your mom and a B allele from your dad. Your blood type is going to be AB. So even though there might be more than one kind of allele for a gene in a population, each individual can only have 2 copies because you only have 2 homologous chromosomes. So here's a visualization of that. So you have possible pairings of homologous chromosomes. Remember, homologous means same genes, different alleles. So homozygous dominant, homo means same, and zygous means zygote. So it refers to the moment of fertilization. So this individual got 2 copies of the yellow allele. This individual got 2 copies, 1 copy of the yellow allele and 1 copy of the green, but the seed outcome is yellow. Curious. This individual got 2 copies of the green allele, and its seeds are green. So heterozygous, hetero means different. This means that within the zygote, there's 2 different copies of the allele for this trait, of the alleles for this trait. And homozygous means same allele. Now we have homozygous dominant and recessive. So dominant traits are ones that mask recessive traits. So in genetics shorthand, we sort of abbreviate using 2 rules. So the dominant alleles are always in uppercase, and the letter of the allele is the first letter of the dominant trait. So the reason I'm doing big Y equals yellow and little y equals green, like why wouldn't I just use a G for that, is because we always pick the first letter of the dominant trait. So as you're practicing with your Punnett squares and some such, just keep that in mind so that when you arrive at the exam, you can execute this skill effectively. So now it's same slide again, just we've superimposed the genotype of the individual in the abbreviation style I just described over the chromosome. So this individual has big Y, big Y. Heterozygotes have big Y, little y. And homozygous recessive individuals have little y, little y. So a full genotype for P color will have 2 alleles, 1 on each homologous chromosome. And as you can see here, in this particular scheme, which we call complete dominance, if there is even 1 copy of the big Y present, the outcome is that the P will be yellow. So even if there is a single recessive copy of the allele present, it doesn't mean that the phenotype is going to be mixed. We'll get to that later. So now we get to ratios. So there's 3 possible genotypes. This one, or this one, but only 2 phenotypes, and Mendel noticed this, and this was significant to him. So this is our 3-to-1 ratio of yellow to green peas. And here's another way to look at it. So this introduces the idea of a Punnett square, which is a way to predict the outcome of a particular cross. So here we have our 2 true breeding plants in the parental generation. So this one has yellow peas and yellow peas always, and this one has green peas and green peas always. So the homologous pairs split during meiosis, and this is our cross. So notice each square contains 1 allele from one parent and 1 from another. And if you cross a homozygous recessive individual with a homozygous dominant individual, what you get are 4 offspring, each of which gets 1 copy of the dominant gene, or allele, and 1 copy of the recessive allele. So that results in an F1 generation with all yellow peas, but the trait for green peas is hidden inside of each of these little eggs. So just a reminder that the alleles split during meiosis. So in this case, instead of using a lowercase y, we're using a grayed out y, so that equals green. And so independent assortment determines what the outcome looks like. But you notice we'll have 1, 2, 3, 4 gametes, and of those, 2 carry the lowercase y and 2 carry the uppercase y. So, homologous pairs split during meiosis. Specifically, what stage of meiosis? Anaphase 1. So it's always important to be tying what you're learning back to what you already know. Okay, so he then allowed the first filial generation to pollinate, and here we have another Punnett square. So in this case, we have two heterozygotes, so one big Y and one little y, contributing to each box. And if you do this, you'll notice you get one homozygous dominant individual, two heterozygotes, and one homozygous recessive individual. So the genotypic ratio is 1 to 2 to 1. But the phenotypic ratio is 3 yellow to 1 green. So I was thinking ahead to evolution. Mendel was only able to count seeds. He couldn't see the genes or the gametes or anything like that, or at least their genotype. All he was able to do was perform controlled crosses and count the relative numbers of different kinds of outcomes. So he determined this genotypic ratio just by observation alone, so just from counting and recording numbers and observing ratios, which is pretty impressive considering. Okay, so you've seen a couple Punnett squares by now, and you should have some idea of how to work through them. So these are used to visualize a genetic cross. So they're basically a tool that helps you graphically make sense of what you can expect to see in a filial generation. But it doesn't give you the outcome as far as numbers of offspring. It just gives you the probability. So yes, each Punnett square has 4 squares. That doesn't mean that the cross will produce only 4 offspring, but if we only have 2 alleles to work with, the box must necessarily contain enough spaces for 1 allele from each parent and 1 from another. So let's try this out using pea plants. So I'm going to say the female gametes, this is a green peed plant, and the male gamete was from a heterozygous yellow peed plant. Okay, so these are the alleles that split during anaphase 1. And in this case, the female gamete was Y, little y. So in this box, we take the big Y from the male and the little y from the female, and same over here. And then down here, we do the same thing. And so our genotypic ratio is 1 to 1 heterozygous to homozygous recessive, and also 1 to 1 yellow to green. So depending on how you perform the cross, the ratios will change, right? And why am I doing 1 to 1, not 2 to 2? Well, because 2 to 2 divided by 2 equals 1 to 1. So we're just simplifying the expression, just as you would in math. Okay, so I've already explained these pieces of terminology, but repetition's our friend when we're just learning things. So, an organism that has 2 identical alleles of a gene for a given character is said to be homozygous for that character. And they can either be homozygous dominant or homozygous recessive. An organism that has 2 different alleles for the same character is said to be heterozygous for that character. And these individuals we call heterozygotes. I'll put quotes around that. There we go. So dominant is the trait that swamps out the recessive trait in the heterozygous condition. So even if you have, say, a lowercase y allele present, that allele is not expressed by the plant, so you don't see any green peas because the yellow condition is dominant over the green. Recessive is whichever trait is not expressed in the heterozygous condition. So the green color of peas is absent in the capital Y, little y condition. It's important to note, however, that the dominant trait is not always the most common or the most healthy, and the recessive trait is not always the least common. So dominant and recessive traits in humans have been summarized here. And although things like migraines or high blood pressure are dominant traits, they're not the most common trait in humans. Now you may be wondering, well, why is that? If dominant always masks recessive, wouldn't dominant genes be more common? And the answer is, well, you might think that, but that would assume that no natural selection was acting on any of these traits, and it absolutely does. When we get to the evolution PowerPoint, I'll explain more about that. But think about this. Even if dominant trait is high blood pressure would predispose an individual to an earlier death, would they not? So, over evolutionary time, it's likelier that individuals with normal blood pressure had better reproductive success and longevity, which means that the recessive trait, although it is recessive, would end up being more prevalent in the population of humans. Okay, so critical result here. Mendel realized that it was possible for organisms to have identical phenotypes but different genotypes, and this was a big deal. So he, just from counting and creating ratios, was able to say, if I'm holding 2 yellow pea plants in my hand, or yellow peas even, it is possible that although they are both yellow, they both appear to have the same character, they can have different underlying genotypes. So far, we have showed you rules for crossing things and only for complete dominance, not incomplete dominance and not codominance. So you can play around with different styles of dominance, and these do exist. So I'll give you some examples. This one is incomplete dominance. So in incomplete dominance, heterozygous individuals, instead of showing the dominant phenotype, show an intermediate or blended phenotype. So in this case, pea plants with red flowers, true breeding red pea plants have capital R, and true breeding white pea plants have little r. Now, if this was complete dominance, you would expect heterozygotes to be red also, but here they're not. So in a first filial generation, F1 generation, we get instead pink pea flowers. And these are capital R. So now we have a 1 to 2 to 1 genotypic ratio, so 1 dominant homozygote, 2 heterozygotes, and 1 recessive homozygote. But we also have a 1 to 2 to 1 phenotypic ratio. So now with incomplete dominance, the genotypic ratio matches the phenotypic ratio. So 1 to 2 to 1 of genotype and 1 to 2 to 1 red to pink to white. Another option for dominance styles is codominance. So instead of getting a blended phenotype where there's sort of an intermediate thing, you get no recessive alleles at all. So whatever is there is expressed, and whatever isn't there is not. So all alleles are equally dominant, and they are just expressed together. So for example, here we have genotypes. So someone with type A blood is AA or OO. I'm trying to draw here, and it's not working. So these are AA or AO. So O denotes no functional allele. So notice in the O condition, there just aren't any surface glycolipids at all. It's not that there's an O-type surface glycolipid, it's just that it's absent. So if you're AA or AO, you have type A. If you're BB or BO, you're type B. And if you have one copy of each, A and B, you're both. So multiple alleles and polygenetics. So Mendel got lucky, in a sense, because he was able to pick an organism that was simple enough that most of its most obvious traits were not polygenetic. So he had 2 alleles to work with, which is how he was able to observe what he was able to observe. So some traits were controlled by multiple alleles of the same gene, and if he had picked something like that, he would have had much more confusing results. So blood type is controlled by multiple alleles, so there's the ABO group of alleles, and there's also the Rh factor, which gives you the plus or minus. And these are separate genes, so they come together in total to tell you what your blood type is. But each individual can only have 2 of the alleles. So most traits are controlled by multiple genes in addition to multiple alleles. So height, eye color, and hair color, these variables are continuous, meaning instead of having a 1 to 2 to 1 ratio, you would get a continuum where, like, if I lined everybody in class up from shortest to tallest, you wouldn't have, 7 people that were 5'2" to 14 people that were 5'8" and 7 people that were 6'1". That's not how that works. Instead, you would see a gradual increase from less tall to more tall. Epigenetics, so epi means superficial to or on the surface. Epigenetics are anything that changes the way a gene is expressed without altering the content of the gene itself. So that's what on the surface of refers to here. Epigenetics means there's no change to the sequence of the DNA. So no change to genetic code. But you can turn genes on and off by doing a variety of things to them. So you can put chemical markers on them to up or down regulate their expression. So an example of this is methyl groups, CH3. If you glue one of these to DNA, it effectively turns off gene. Or you can also do things like wrap DNA more tightly around a histone. So these can be affected by environment, nutrition, development, a variety of things. And it turns out that to some extent, the epigenetic tags are a little bit heritable, which we'll get to in the next slide. So genes can be either completely turned on or off, or they can simply be enhanced or reduced. So in the next class, we'll recapitulate some codominance and incomplete dominance a little bit, and we'll also talk about the inheritance of genetic disorders as well as the mechanisms of inheritance for epigenetics. But for now, I'm going to leave it here. So thank you for your attention and for your patience in waiting for this to come out, and I will see you in the next video.